A wild dromedary lowers its head into the salt mist before dawn and crops mangrove leaves off the Arabian shore. A camel living a tidal forest, sea spray life that nobody picturing the animal today would ever imagine. It is 6,000 years ago, on the coast of what is now Oman, and this creature is doing something that will turn out to matter enormously. It is tall, pale, long-legged, built for thirst and salt and distance. And in a few thousand years, every member of its kind living free in the world will be gone. We call its descendants camels. They became the engine of the incense trade, the ships of the Sahara, the animal that made the Silk Road possible across stretches of sand where no horse could survive. And for a very long time, we thought we understood where they came from. Nobody photographs that wild herd. Nobody records how many there were, or how far they ranged, or what finally drew the first of them toward human fires. We only know that this exact creature, foraging in the coastal mangroves of southeastern Arabia, sits at the root of a story that would carry humans and goods across the greatest deserts on the planet for the next 4,000 years. We were wrong about almost all of it. We were wrong about where they were tamed, wrong about which wild animals their ancestors were, wrong about whether the wild original even still exists. The camel kneeling in a market in Cairo or running in a race in Dubai is not the simple desert native of the textbooks. It is the survivor of a vanished wild lineage, a creature whose true origin was hidden until scientists learned to read DNA out of 7,000-year-old bone. This is what the DNA actually shows. For most of the last century, the camel's origin was treated as one of those facts too obvious to question. There are two kinds of camel in the Old World. There is the one-humped dromedary of Arabia and North Africa, and there is the two-humped Bactrian camel of Central Asia. The names themselves seem to settle the geography. The dromedary belonged to the Arabian deserts. The Bactrian belonged to Bactria, the ancient region spanning what is now Afghanistan, Turkmenistan, and it took its very name from that homeland. The evidence behind these assumptions was real, and it deserves to be taken seriously. The dromedary was an Arabian animal in every way that mattered to the people who lived with it. Its body was a desert machine. It could lose a quarter of its body weight to dehydration and survive, drink 100 liters in minutes, close its nostrils against blowing sand, and walk for days across terrain that would kill almost any other large mammal. Its red blood cells are oval rather than round, so they keep flowing when the blood thickens with dehydration and do not burst when the animal floods its body with water at a trough. The fat in its hump can be metabolized for energy when food runs out. Arabian rock art and early inscriptions showed camels. The oldest dense concentrations of camel bones in human settlements came from the southern Arabian Peninsula. If you wanted to know where the dromedary was first tamed, you looked at the place where it was most useful, most celebrated, and most embedded in human life, and that place was Arabia. The Bactrian story looked even tidier. The two-humped camel carried the name of a Central Asian kingdom. Archaeological remains of two-humped camels turned up across the region between Iran and China. The animal was woven into the trade networks of the Silk Road, hauling silk and lacquer and tea across the cold, high passes, where it could shrug off temperatures that swung from blistering summer to 40 below in winter. It seemed natural to assume that the place it was named after was the place it had been domesticated. Each theory had its evidence, each had its logic, and each rested quietly on the same two flawed assumptions. The first was that the place where an animal is most associated with human culture must be the place it was first domesticated, when in fact an animal can be tamed in one region and become iconic in another. The second, and more dangerous, was the belief that you could find the homeland of a domestic animal by studying the genetics of the living population. For decades, that was the only DNA available. Researchers sampled modern camels, looked for the region with the greatest genetic diversity, and expected it to point back toward the origin like a compass needle. With dromedaries, that needle simply spun. The problem was hiding inside the dromedary's greatest strength. This was the animal humans relied on to move goods across the largest trade networks of the ancient world: incense up the Arabian coast, silk and salt across the Sahara, caravans threading between the Middle East and North Africa for thousands of years. And that constant movement did something to the camel's genome that almost never happens in a domestic animal. It mixed it. Caravan camels from distant regions met, bred, and exchanged genes again and again, century after century. The result was a species with almost no geographic structure in its DNA at all. A camel from Morocco and a camel from Pakistan can look genetically like neighbors. The trade routes had stirred the gene pool so thoroughly that the homeland was erased. Reading modern dromedaries to find the first dromedaries was like trying to find the source of a river by studying the ocean it empties into. Consider how different this is from almost every other domestic animal. Cattle, pigs, goats, sheep, each tends to carry regional genetic signatures, clusters that betray where local herds were kept and bred in relative isolation. Wander across Europe sampling cattle, and the DNA sorts itself into neighborhoods. Do the same with dromedaries from Senegal to Rajasthan, and the neighborhoods dissolve. The camel was simply too good at its job, the very thing that made it valuable, its ability to walk thousands of kilometers and arrive ready to breed, guaranteed that no population stayed put long enough to develop a distinct genetic identity. The animal erased its own paper trail by being too useful. The only way forward was to stop reading living camels and start reading dead ones, the bones of wild and early domesticated animals that had died before the trade routes blended everything together. That is exactly what one of the most ambitious camel studies ever attempted set out to do. A large international team, coordinated from the University of Veterinary Medicine in Vienna and published in the Proceedings of the National Academy of Sciences, set out to find the dromedary's homeland by brute force. They sampled nearly 1,100 living dromedaries from across the entire range of the species, from West Africa to South Asia. Then they did the thing that mattered most. They extracted DNA from archaeological bone, the remains of wild and early domesticated dromedaries, some of them up to 7,000 years old, recovered from sites scattered across the Arabian Peninsula and beyond. Extracting usable DNA from desert bone is its own quiet miracle. Heat is the enemy of ancient genetics, and the Arabian climate is about as hostile to molecular preservation as any on Earth. The researchers worked from the densest, best protected fragments, the petrous bone of the inner ear among them, to pull readable sequence out of material that by every expectation should have been blank. They compared the ancient genetic signatures against the modern ones, searching for the place where the deepest, oldest diversity survived. The answer that emerged was specific in a way the modern data alone could never have delivered. The dromedary was domesticated on the coast of the southeastern Arabian Peninsula, in the region of modern Oman and the United Arab Emirates, around 3,000 to 4,000 years ago, not in the deep interior deserts, on the coast. The textbook had pointed vaguely at Arabia, and for once the broad direction was right. But the real story was narrower, stranger, and tied to a landscape almost nobody associates with camels: the salt marshes and mangroves of the Arabian shore. The first revelation was that the wild dromedary had been a coastal animal. The archaeological evidence pointed to wild dromedaries that foraged in coastal habitats, feeding among mangroves and salt-tolerant plants. This was not a creature of the open dune sea that we picture today. Salt is essential to a camel's health, far more than for most mammals, because an animal built to conserve water still has to flush mineral loads through its system. and the coast offered it freely, in the sea spray, in the halophyte vegetation, in the brackish margins where the land met the Arabian Sea. The gray mangrove that lines this coast, Avicennia marina, sheds excess salt through its very leaves. An animal browsing it is licking the sea out of the foliage. The creature that would become the ultimate desert traveler began as something that browsed the tidal forests of a vanished coastline. Domestication did not happen where the camel was most at home in the desert. It happened at the edge of the water, where the wild herds and the coastal human settlements overlapped. Fishing communities, shell-midden people, the kind of settled coastal life that leaves bone and hearth behind in the sand. But here is where the story becomes strange. When the researchers looked for that wild dromedary today, the coastal browser, the salt marsh forager, the original animal, they could not find it. It does not exist. The wild dromedary is extinct. Every dromedary alive on Earth today, all the tens of millions of them, is a domesticated animal, or the feral descendant of domesticated animals. The large feral herds running loose in the Australian outback, descended from animals shipped in during the 19th century to haul supplies across a continent, are sometimes called the only truly wild dromedaries left. But they are not wild in the deep sense at all. They are escaped livestock, the children of tame stock gone loose, not survivors of the original lineage. The creature that walked out of the mangroves to meet the first herders left no free-living lineage behind. We did not just tame the wild dromedary, we outlived it. The ancestor is gone, and only its altered descendants remain to tell us it was ever there. And here, the story becomes darker, because the Bactrian camel carried a parallel ghost. The two-humped camel was the one that seemed safest of all. After all, a genuinely wild two-humped camel still survives today, a critically endangered animal clinging to existence in the Gobi Desert of Mongolia and China. Fewer than 1,000 of them remain scattered across some of the most remote terrain on the planet. Animals so hardy that one population survives by drinking water saltier than seawater from desert springs. For a long time, it was natural to assume this wild camel was the direct ancestor of the domestic Bactrian, the living root of the lineage. Combined ancient and modern DNA analysis demolished that assumption. The wild two-humped camels of Mongolia and China are not the direct ancestors of the domestic Bactrian camel. They are a separate lineage, a distinct branch that split away long before domestication and survived in parallel, not the source population at all. The split runs deep, hundreds of thousands of years deep, far older than any herder. The true wild ancestor of the domestic Bactrian, like the wild dromedary, appears to be gone. The last wild two-humped camel on Earth is not the camel's living grandfather. It is a distant cousin from a branch that simply happened not to die. So the eponymous homeland fell too. The two-humped camel that took its name from Bactria was not, the genetic and archaeological evidence suggests, domesticated in Bactria at all. The originally assumed center of origin in the region of modern Afghanistan and Turkmenistan has been replaced by probable domestication centers further west, in Western Asia, between 5,000 and 6,000 years ago. Roughly 1,000 to 2,000 years before the dromedary was tamed. The name on the animal pointed in the wrong direction. The Bactrian camel was not born in Bactria any more than the dromedary was born in the dunes. Both animals carried a homeland in their names and in their reputations that the DNA quietly overruled. The DNA cannot tell us exactly why the wild ancestors vanished. It can only tell us that they did, that two separate wild camel lineages on two different continents gave rise to the world's domestic camels and then disappeared from the wild, leaving their tame descendants as the sole inheritors of the line. Whether they were hunted out, absorbed through interbreeding with domestic herds, or squeezed into extinction as the climate and the human world closed in around them, the result is the same. The wild original is a genetic ghost, readable now only in the bones we dig out of the ground. There is a grim logic to how it might have happened. A coastal, wild herd, already living in a narrow ribbon of habitat between desert and sea, would have been easy to hunt, easy to crowd out, and easy to absorb. Once tame herds were grazing the same salt marshes. A wild population does not need to be slaughtered to vanish. It only needs to be slowly outnumbered until the last unmixed animal dies and no one notices which one it was. There is one more genetic strangeness the dromedary study uncovered, and it reaches back to that problem of the spinning compass needle. When the researchers examined the modern population, they found that the dromedary's genetic diversity was directly shaped by its use as a transport animal. The constant gene flow between distant populations, the kind of regular mixing you almost never see except in wild species, was the fingerprint of the caravan. The trade routes had kept the species genetically unified across thousands of kilometers, blending West African camels with Arabian ones with South Asian ones, generation after generation. Only a single population stood apart from the rest, one group that had been isolated for a time by geographic and cultural barriers, drifting away from the shared diversity of the species. The dromedary's genome, in other words, is not a record of its biology alone. It is a map of human commerce, every merchant route and pilgrimage road written into the animal's DNA. You can read the Hajj in a camel. You can read the frankincense road in the spread of its genes. The story does not end in the old world because the camel family has a second, even older secret on the far side of the planet. Long before any of this, the deep ancestors of all camels arose in North America and spread out across the world. For most of their history, camels were an American animal, small early forms grazing the plains of what is now Nebraska and Texas tens of millions of years ago. One branch crossed the Bering land bridge into Asia and Arabia to become the camels we know. Another branch went south, across the Isthmus of Panama, into the Andes, and became something else entirely: the llama, the alpaca, the wild guanaco, and the vicuna. The original North American camels then vanished completely, dying out at the end of the last ice age and leaving the family to survive only at its two far-flung extremes. These South American camelids carried their own buried history, and ancient DNA has been quietly rewriting it too. The accepted account held that the wild guanaco was domesticated into the llama and the wild vicuna into the alpaca in the high Andes around Lake Titicaca and Peru some 6,000 to 7,000 years ago. The broad outline survived, but a genetic study published in eLife, drawing on ancient DNA from camelid bones at the Tulan archaeological sites in the Atacama Desert of northern Chile, one of the driest places on Earth where preservation is so good that organic material can survive for millennia, found a now familiar pattern hiding underneath. A substantial proportion of the ancient vicuna genetic variation has been lost. It simply is not present in any living animal, and the researchers proposed that one of the founding domestic populations descended from an ancient guanaco population that no longer exists. The same ghost haunts the Andes that haunts Arabia. The wild founders of the domestic camelids were not preserved intact in their living descendants. They were partly erased, surviving only as genetic echoes in old bone, a separate parallel companion study reaching the same unsettling verdict from the opposite hemisphere. Two hemispheres, two teams, two sets of ancient bone, and the same answer: the wild animal at the beginning is missing. For most of camel history, none of this would have made any difference to the animals themselves, because for thousands of years, a camel was a working animal shaped by the desert and the road, not by human aesthetic design. Its body was sculpted by the demands of the caravan: a strong back for cargo, padded feet for sand, fat stored in the hump against famine, a tolerance for thirst that bordered on the miraculous. There were regional types: the lean, fast-riding camels of the deserts, the heavier baggage animals of the trade roads. But there were not breeds in the modern sense. No closed stud books, no show standards, no pedigrees traced for prestige. The selection pressure was simple and brutal. An animal that could not carry a load across 100 waterless kilometers did not pass on its genes. Then, in the last stretch of the story, that changed. In the Arabian Gulf in particular, the camel has been transformed from a beast of burden into an object of competition and luxury within living memory. Camel racing has become a high-stakes sport, with elite racing dromedaries selectively bred for speed and worth staggering sums, run on dedicated tracks where robotic jockeys now ride in place of the children once used. Camel beauty contests now award fortunes for the precise shape of a head, a neck, a lip, a hump, to the point that some entrants have been disqualified for cosmetic enhancement, for Botox injected into the lips, for surgically inflated features, for hormones. A working animal that spent 4,000 years being shaped by sand and distance is now, in some quarters, being reshaped by the show ring and the racetrack, its genome bent toward the aesthetic ideals of the last few decades. The deep camel, the salt marsh forager turned desert ship, still lies beneath. But laid over it now is a thin, glittering layer of very recent human invention. What does this mean for the camel kneeling in a market or pacing a paddock right now? It means the animal in front of you is the last living chapter of a story whose opening pages have been torn out. Its wild ancestor, the coastal browser of the Arabian mangroves, or the vanished guanaco of the Andes, is gone from the earth, surviving only as fragments of DNA in ancient bone. The dromedary was tamed not in the deep desert, but on a salt marsh coast that has since changed beyond recognition. The Bactrian was not born in the Bactria that named it, and the living, wild, two-humped camel of the Gobi, that last free herd, is not the ancestor we long assumed, but a cousin from a branch that merely escaped the same disappearance. The camel is not a simple desert native. It is a genetic ghost story with humps, a domesticated survivor carrying the memory of wild lineages that no longer exist, its very genome written over by 4,000 years of human trade. The familiar animal at the edge of the oasis is far older and far stranger than the textbook ever let on. The fog still comes off the Arabian Sea before dawn, the way it did 6,000 years ago. But the mangroves are quieter now, and the pale wild herds that once moved through them are gone. We do not know which animal first walked toward the human fires. We do not know which night or which coast or which herder first reached out a hand. We only know that whatever happened there, at the edge of the salt water, set in motion an animal that would cross every great desert on Earth, and that the wild creature who began it all vanished into the ground, leaving only its bones and its tame descendants to remember it ever lived. If you found this story remarkable, consider subscribing. There are more origins to uncover, and the DNA is only beginning to tell us what it knows.